Bismillah. Assalamualaikum warahmatullah. You're listening to microbiology lessons on acellular microorganisms. We're going to cover three things as the learning outcomes. Number one, you want to be able to discuss the features and physical chemical structures of viruses, meaning what are the components of viruses and how they differ from bacteria. Secondly, we'll talk about how viruses replicate. I'll give you some typical variations of it so you'll learn the general pattern of how it works. And number three, we want to distinguish between viruses, viroids, and prions. Let's start with the features of what makes something a virus. So the first traditional feature is that they are obligate intracellular parasites. What that means is typically viruses can only grow in live host cells. Now, I said typically because as you learn deeper, you realize that it's not always the case. For example, I read a paper where people have successfully synthesized a virus, a T4 virus, using what's called TXTL system. What that means is you don't need live cells to grow that virus. You just need cell lysate components for transcription and translation, and then you can synthesize viruses from that. So, outside of this lecture, you might hear people say viruses are obligate intracellular parasites, that they must grow in living cells. When you hear that, keep in mind that it's not 100% true. But of course, don't argue with them. That's an important spiritual lesson in immunology and microbiology education. As you become more advanced in science, you're going to notice more errors in others. But don't point out how wrong people are unnecessarily. Be kind. All right. Viruses have host range and specificity. Host range means how many species can the virus infect. For example, poliovirus can only infect humans and maybe some primates, but it won't infect birds or other avian species. Viruses are also very picky, very specific about their cellular targets. For example, SARS-CoV-2, the COVID virus, can infect lung cells, but not kidney cells. The second feature is viruses are often much smaller than bacterial cells. So imagine you are of the size of virus. Yeah. So to you, a human liver cell is like a massive jumbo aircraft that can carry hundreds and hundreds of you. A bacterium like E. coli is around, I don't know, 3 microns, 3000 nanometers. So the bacterium looks like a big bus to you as a virus. So viruses are smaller than bacteria. The third feature is viruses usually have DNA or RNA, but not both. And again, I say usually because I've read about a DNA RNA hybrid virus. They found it in, I think, California, some volcanic park. But most viruses only have either DNA or RNA. The fourth feature is their nucleic acids are surrounded by capsid, not plasma membrane, not like bacteria. And finally, they don't generally multiply outside of their host. They need the host protein synthesis apparatus. When you compare viruses and bacteria, you're going to see crucial differences. Bacteria typically are not intracellular parasites, but viruses are obligate intracellular parasites. Bacteria also have both DNA and RNA, but viruses only have either of the two. Bacteria propagate using binary fission. Viruses don't do that. We'll talk about how they replicate in a minute. And this one is important. Bacteria can be filtered by bacteriological filtration in the lab. But if you use that to filter viruses, it won't work. The viruses will just pass through it because they are way smaller. Bacteria can generate their own adenosine triphosphate, ATP, their energy molecules. Typical viruses don't do that. Bacteria have ribosomes. Typically, viruses don't. Viruses have to use the host ribosomes for synthesizing proteins. And if you get infected with bacteria, the doctors can treat you with antibiotics. But when you have viral infections, antibiotics don't work. The doctors will instead give you some sort of broad spectrum interferons. So those are some of the differences. Viruses also differ from bacteria structurally. So deep in the middle of a virus, you have your nucleic acids, which are packaged inside a capsid. The capsid is surrounded by viral envelopes, and on that membrane envelope, you have protein spikes. So The specificity of viruses comes from these spikes. They can only stick to specific receptors on specific target cells. Nucleic acids in viruses can be either DNA or RNA. They can also come as single-stranded or double-stranded strains. Double-stranded viruses are, for example, hepatitis B, which can cause liver disease. An example of single-stranded virus is, SARS-CoV-2, COVID virus. Viral nucleic acids can also have positive sense or negative sense. And if you remember from your genetics class, this of course will affect the number of steps in their replication. Because SARS-CoV-2, for example, is a positive sense virus. So that means its RNA can be translated by the host cell's ribosomes directly. That leads to quicker protein synthesis. It means the virus can replicate faster. An example of negative sense viruses is influenza viruses. They need an additional step. The RNA must first be converted into positive sense and then it gets translated. Those nucleic acids are surrounded by capsids. The subunit of capsids, we call them capsomeres. So we put capsomeres together, we'll get capsids, all right? So not the other way around. Don't confuse the two. The way we put capsomeres together will determine the shape of your viruses. So they can be icosahedral, helical, and a lot of other shapes. The shapes of the viruses are useful for diagnosis, for identifying what kind of virus a patient is infected with. And actually, the shapes of the viruses influence their pathogenicity as well. So viruses that are filamentous, for example, elongated, they sometimes use their shape to bridge the gap between cells. So it helps them infect the next cells. What else? Those capsomeres, they don't have to be of the same protein type. Yeah. So some viruses do have capsids with capsomeres that are made up of different types of proteins. Many viruses are coated by viral envelopes. So these viral envelopes are actually the membranes of the host cells. The viruses took the membrane when they killed the cells. So if you want to anthropomorphize that, it's rather horrific. It's like a pirate killing a victim and then taking the skin of the victim and wearing the skin. But not all viruses are that gruesome. Some viruses don't take their victim's membrane. They just burst out of the cells and leave them dead. So the viruses that are not wearing the membrane envelopes from the host cells are called naked viruses. So they don't have envelopes. On both of them, you have spikes. You have spikes on these envelopes. If you are naked, you have spikes on your capsids. Spikes are used as binding receptors to the target cells. So that's the reason why viruses have specific hosts. It's because their receptors only recognize certain configurations on the host cell's membranes. So only if it matches, then the replication process will activate. So those are generic structures for all viruses. Now, let's see some specific examples, yeah? I'll give you two examples, influenza and bacteriophage. If you look at the structure of an influenza virus here, at the core of it, you're going to find a ribonucleoprotein complex. You see, in certain viruses, the nucleic acids don't just float around by themselves. Sometimes they are bound together with proteins. So here, the RNA is bound to RNA binding proteins to form what we call ribonucleoprotein complexes, or RNP. These complexes are surrounded by a layer of capsid, a thin layer of capsid, which is then surrounded by a membrane envelope. On that envelope, you have your spikes. For influenza, those spikes are called hemagglutinin and neuraminidase. So when you hear about influenza viruses, you're going to hear about like H1N1 and H3N2, for example, right? So that naming convention refers to the spikes. It refers to the types of hemagglutinin and neuraminidase that the virus has. So H1N1 means the virus has hemagglutinin type 1 and neuraminidase type 1. H3N2 means the virus has hemagglutinin type 3 and neuraminidase type 2. So those are the spikes. The further you study virology, the more components you're going to see. An example of that is the M2 ion channel for influenza. It's for proton conductance. It transports protons across the viral envelope into the virus because influenza needs that acidity to release the ribonucleoprotein. Our second example is bacteriophage. When I was younger, the first time I saw this, it looked like an alien creature. I mean, look at it, right? It has a head and tails. So for bacteriophages, the nucleic acids are inside the viral head and not a capsid. And that viral head is attached to a collar, like its neck. So it connects the head to a long tail sheath, which can contract. At the bottom of the tail sheath, you have the plate with pins on it, and these pins are used to penetrate the host cells. Bacteriophages don't have spikes. Instead, they have these tail fibers that looks like legs, right? So one question is, how do they attach to the host cells without spikes? Well, they use these tail fibers. Bacteriophages target bacteria, by the way. Bacteriophage etymologically means bacteria eater. So they specialize in attacking bacterial cells. So once they are attached successfully, they will initiate replication. Since we just talked about bacteriophages, I'll give you the replication for phages first. And then I'll give you another two variations for other viruses. So you will learn just three basic examples of viral replication for this course, all right? Let's start with bacteriophages. This is your sequence of events for phage replication. We call this the lytic cycle. Firstly, we have adsorption. The phage binds to the exterior of bacterial cell. Think of this as a key entering a lock. So it's a highly specific interaction. A specific phage will bind to only specific bacteria. Then you have penetration. The phage breaches the cell wall and membrane of the bacterium, and here it injects its DNA. It's like inserting a new set of instructions into the cell's computer to reprogram the computer. And part of that reprogramming will disrupt the bacterial own DNA replication. Next in the cycle is biosynthesis. The phage DNA takes over the cell's metabolic pathways. It redirects the bacterial machinery to produce viral components. So the cell is now working for the enemy. The cell then assembles the building blocks of the new phage particles. The fourth stage is maturation. This is similar to an assembly line in a factory. So you have the new phage components come together to form complete phages inside the bacterial cell. And finally, it's the release. The bacterial cell lyses and that releases the mature phages. And these phages are now free to infect adjacent bacterial cells and start the cycle again. Now, let's look at the alternative to the lytic cycle. It's called lysogenic cycle. First, when entering the cell, the phage DNA integrates into the cell's chromosome. So it forms what we call a prophage. And it's crucial to note that at this point, the phage is not destroying the cell. Rather, it's quietly biding its time. It's lying dormant within the host genome. When the cell replicates, the prophage gets replicated as well. So what happens then is each daughter cell from binary fission now contains a copy of this viral DNA. So this is the crux of the lysogenic cycle, yeah? The phage DNA is propagated along with the host DNA across countless cell divisions. So contrast that with the lytic cycle that we learned just now. In the lytic cycle, the phage also takes over the cell's machinery, but the phage doesn't do that to coexist quietly. It does that to multiply and to cause the cell to lyse. Now, in the lysogenic cycle, it can be interrupted. We call that induction. The induction triggers the dormant prophage to initiate the lytic cycle. So in this sense, you can think of the lysogenic cycle like a ticking bomb. It waits for the right moment to switch from the lysogenic phase to the lytic phase. Now, this one is the replication of a DNA virus. So the cycle also begins with adsorption, where the virus binds to a host cell. It's specific, it's targeted. The viral envelope then merges with the cell membrane. We call this fusion. Next is penetration. The virus enters the cell and the viral DNA is released from its capsid. So this step is referred to as uncoating. Once inside, the viral DNA moves to the cell's nucleus. Now, here, the transcription takes place. The host cell's machinery is co-opted to produce viral mRNA. They don't write the mRNA here. This mRNA is then used in the translation process. So the cell's ribosomes read this mRNA and begin making viral code proteins. And meanwhile, viral DNA is replicated and new viruses are assembled from these components, and this is maturation, and finally release. New viruses exit the host cell. Some wrap themselves in a piece of the host membrane, creating an envelope, and now they can infect new cells. This one is the replication of an RNA virus. Influenza virus is a negative sense RNA virus. The cycle starts with the virus latching onto the cells. This adherence or adsorptions is step one. The virus then gets internalized by the host cells. Once inside, in the endosome, the virus encounters acidic environment. The virus then uses the M2 ion channel that I mentioned before to trigger the release of viral RNPs, ribonucleoproteins. The RNPs are then released into the cytoplasm, and these RNPs carry the virus's genetic blueprint, so they must reach the cell nucleus, so that's their target. Inside the nucleus, the virus uses the host machinery to create mRNAs, messenger RNAs, and cRNAs, complementary RNAs. And then here's what happens. The mRNAs exit the nucleus and instruct the cell to produce viral proteins. At the same time, the cRNA act as templates. They guide the formation of new negative sense RNAs. So these RNAs then become part of the RNP of the next generation of viruses. The final stage is assembly and release. All newly made viral components converge at the host cell membrane, and then the new viruses break free. They exit the cell and ready to continue the next cycle of infection. All right, so those are the three variations of replications. Now, no matter which replication process is occurring, they end up with some of these three outcomes. The first is lysis. As I explained earlier, the virus grows, breaks the cell, and moves on. It's a messy but effective way to spread. We see it in both bacteriophages that infect bacteria and viruses that infect animals like influenza. The second possible outcome is mutation. So here's the deal with viruses, especially the RNA viruses. They don't copy themselves perfectly, so they lose fidelity. Each time they replicate, they make errors. And that leads to new variants of the virus, new versions of the virus. So the variants can become more dangerous than the ones that existed before. The third is apoptosis. Some viruses can make the infected cells kill themselves. So that process is called apoptosis. HIV does this to some immune cells, which is why HIV is so lethal to the immune system. Now, we can use the replication process to cultivate viruses for experiments. There are loads of methods for cultivating viruses, and we can generally categorize them into two, in vivo and in vitro. In vivo means inside living organisms. So, for example, if you cultivate your virus in mice, that's an in vivo experiment. Immunologists, for example, use the in vivo experiments because it mimics the natural environment. For instance, they would look at the immune response to a lung infection in mice. So that is more likely to be closer to how human lungs respond to the infection compared to experiments on a petri dish, which also works actually. Now, the experiment on petri dishes is in vitro. In vitro means outside living organisms. So if you grow your virus in cell cultures instead of animals, that's in vitro. We choose in vitro to simplify our experiments so we can focus on one variable at a time, one pathway at a time, one aspect at a time. We also use in vitro to save costs. It's cheaper than animal studies and it's faster to do, broadly speaking. We use in vitro, for example, to measure the amount of virus. So we do what's called plug assays. You mix your virus sample with cells in a medium, then you incubate it, then you'll see plugs, the clear spots on your petri dish. The more plugs you see, the more viruses you have. All right, we're done with viruses. Next, we're going to look at acellular microorganisms that are much simpler than viruses. They are called viroids. Viroids consist of RNA particles. So this isn't the RNA we find doing regular jobs in a cell. It's a pathogen. So it's simple and it's dangerous. Viroids are just RNA, infectious RNA. They don't even code for protein. Yet they can still hijack cellular replication systems. Viroids infect plants. They don't kill the plant outright. Instead, they disrupt growth, so the yield of the plant goes down. An example of it is the potato spindle tuber viroid. This particular viroid distorts potato tubers, so it makes the potatoes look weird, and it reduces the plant growth and damages its agricultural and commercial value. Viroids differ from viruses in several ways. Firstly, viroids are composed of only a single circular RNA molecule. This is quite distinct from viruses, which can have RNA or DNA and usually more than one molecule of nucleic acids. And the nucleic acids are often linear rather than circular. Number two, unlike viruses, viroids don't have capsids or envelopes. So viruses typically have a protein coat or an additional lipid envelope, which viroids lack entirely. So this absence means viroids are just naked RNA, nothing more. Thirdly, viroids do not produce proteins. Viruses take over the host machinery to produce proteins because this is essential for their life cycle. Viroids don't do that, which is quite an exception in the microbial world. Fourthly, viroids are only replicated in the nucleus of the host cells. This is in contrast to many viruses which can replicate in various parts of a cell, like the cytoplasm. And lastly, viroids can be detected only through special techniques because we need to identify their unique nucleotide sequences. So this is unlike the methods used for virus detection because with viruses, we can identify the presence of viral proteins. Now, look at the blue and purple strands here. So these larger structures are the DNAs of a T7 bacteriophage. And now look at these tiny, tiny yellow rods. So these are viroids, just short RNA strands, very tiny. This is all it takes for them to grow and damage our agriculture. So discoveries like viroids make us rethink what pathogen is and what life forms can be at the microscopic level. All right, viroids are RNAs without proteins, right? Now, prions are the opposite. Prions are proteinaceous particles, meaning they are composed solely of protein. They lack any kind of nucleic acid, no DNA, no RNA to carry genetic instructions. Prions cause diseases such as Creutzfeldt-Jakob disease or CJD in humans and mad cow disease in cattle. They have misfolded proteins accumulated in the brain, leading to tissue damage. Here's what I mean by misfolded proteins. So both of these are prion proteins. So the left one is harmless form of it. The one on the right is misfolded. They look different. And that's the one that causes diseases. And then also this misfolding can cause other normally folded proteins to also misfold when they encounter each other. So that's how prions spread the disease. Prion diseases can happen in many animals, not just humans. So sometimes they can spread from one species to another, and other times they just happen like a random error of protein misfolding without any spread needed. So the point of this figure is for you to understand that the disease can be passed between species. And for humans, the impact is twofold. You have classic CJD, which is like a time bomb, sometimes taking up to 40 years to show symptoms. And then there's the VCJD, a newer form of the disease that can take hold in just 2 to 4 years. So both can come from something as normal as eating the infected meat. All right, this is the last slide. Just like viroids, prions are also different from viruses. Firstly, they can withstand temperatures that readily inactivate viruses, such as 90 degrees Celsius. Secondly, viruses are typically susceptible to radiation. Prions are not susceptible to radiation. Why? Because prions haven't got any DNA or RNA to damage, so radiation doesn't work on them. And for similar reason, prions are also unsusceptible to DNAs and RNAs. So these enzymes break down nucleic acids. Since prions lack DNA or RNA, they are not affected by these enzymes, unlike viruses, which can be taken down by these enzymes. All right, so that's it. You've learned the features of viruses, you've learned various ways viruses replicate, and then you learn two other acellular microorganisms, which are viroids and prions. All right, talk to you next time. Keep on reading, keep on learning. Barakallahu li wa lakum. Wassalamu alaikum wa rahmatullah.